Video 8. This one is genetics and epigenetics with regards to what's going on in the brain, obviously. So if you're thinking that from your unit on applications of genetics, you know that there are just over 20,000 genes that they have found in the human genome. About a third of those genes are becoming expressed, are being turned into polypeptides and proteins in the brain. That's a higher percentage than any of the other human body organs that we know. So a third of the genes are actually expressed. Now, wherever there are genes that are being expressed, it could be that those modifications, the myelination, the histone stickiness, and so on, that is something that could happen to all those genes, whether they're coding for a neurotransmitter or some sort of transport protein. All those epigenetic changes could potentially produce a long-term developmental change in the person that they've had. There is an absolutely fantastic blog that I found on the Dutch famine. I'm hoping that on the PowerPoint you're going to find that blog. Otherwise, just type in Dutch famine and see what you can find. Dutch famine neuroscience. It's an example of epigenetics. Picture mothers while pregnant struggling from undernutrition, literally starving during the actual pregnancy. Now, in order to get as much energy out of food as possible, insulin control is completely different, and how much energy is going to be reabsorbed as soon as there's any food available. That, when do you switch on insulin or the hunger hormones or something is being regulated that would allow as much food as possible getting into the body. That's the appetite controlling regions, probably a polygenic situation, not just one. It's still being researched. There's evidence that the children and the grandchildren of such mothers that have been starving are struggling with obesity and type 2 diabetes much more than others. That's a statistically significant situation here. So I'll leave you to do a bit of extra research. That's a lovely blog, that link here, so if you can find that. Now, the epigenetic link seems to be unique to mammals. But there seem to be particular factors that affect humans much more than others, or maybe that is our perception at the moment. If human babies are particularly underdeveloped at birth, much shaping is happening due to social and environmental factors compared to other mammals. If you think of other babies that are being born, many ape babies and monkey babies know how to cling on to mom. Human babies don't know how to do that. They're literally helpless. So they are relying on their social context and their environmental context for the first few months of survival. They're literally unable to do anything for themselves at the start. There's another little line of evidence from animal care. So think of dogs, for example. If they're stressed very, very early on in their lives, let's say puppies that are being taken away from mom far too early and losing their siblings far too early than they should be. Anyone who's looking into pet care will know about that. That stress may lay down an epigenetics change which silences certain genes, and therefore the stress levels in that animal will be there for life. There's a lot of empirical evidence from animal breeding with that. So what other studies do you have that give you ideas about epigenetic changes? Let's say something genetic is there to cause schizophrenia, depression, anxiety. Shouldn't twin studies show that if you've got identical twins, they have been separated at birth, different upbringings, and would they both still develop schizophrenia or depression or anxiety, even if in completely different environmental and social backgrounds? That gives information. Now, it's not simple at all. We have got polygenic situations, and then on top of it, environmental factors playing into it for these conditions. So not straightforward. This is literally just what lines of evidence do we have, what informations do we have. So if psychiatric conditions, quite a lot of psychiatric conditions have got a polygenic backing, and obviously the polymorphism that you've got maybe 1 or 2 or 3 different alleles coding for the same, or on the same gene. on same locus, then you could have that already as a variable. But also on top of the inherited part, there would be environmental experiences contributing to how things like schizophrenia develop. It's a bit like cancers. You might have an inherited condition that will never break out unless you've got these and these environmental triggers there as well. Now, there are two conditions that are linked to specific areas in the brain and specific chemical abnormalities in the brain. So that is now just literally physiological abnormalities again, and anatomical differences. If you've got a patient who is bipolar, which is a condition where the flicking from happy personality to a sad personality happens quite quickly. That's how I understand bipolar, but I'm not a psychiatrist, so probably I'm not the best person to say what bipolar is. It's linked to an increased volume of an area in the brain called the amygdala. That is one thing. So we think, hold on a second, are psychiatric conditions based on some physical change in the brain? Is that now genetic or epigenetic? Various other schizophrenia, ADHD, OCD, various phobias and post-traumatic stress are all linked to some abnormalities in neurotransmitters. Now, neurotransmitters are mostly protein based, or an enzyme is made available in the production of those neurotransmitters. That means those proteins have to be coded for. What if the genes for those proteins have been silenced? The two things that I've highlighted underneath and made a bit bigger in blue, the substance abuse and the childhood experiences. I'm going to continue with this on the next slide. The substance abuse can cause physical changes to the brain with regards to sizes and with regards to neurotransmitters, as well as synaptic changes. Childhood experiences. There is evidence of children that have been in abused situations are whole loads more likely to suffer with a range of psychiatric conditions, and epigenetic changes are possible causes for that. So taking those apart a little bit, what can you actually evidence in with all versions of imaging? What evidence do we have? For the substance abuse, there is evidence that the myelination is being damaged by alcohol abuse. So the white matter is being damaged by alcohol abuse. What psychiatric conditions arise out of that? Completely different story, different question. LSD and amphetamines are linked to paranoia and anxiety. The actual pathway at this point, as far as I know, is not known yet. Cannabis has been proven to worsen existing depression, not cause it in many cases, but to definitely worsen. That has been proven. Now, statistically, if childhood experiences of abuse are there, then you've got a whole list of psychological, psychiatric conditions that can be caused: depression, anxiety, schizophrenia, eating disorders, personality. The list is long, so there is a link. Whether or not that's epigenetic is still to be shown. So whether or not a childhood abuse silences a gene that would have coded for this neurotransmitter, and therefore, because of the inavailability of that neurotransmitter, something else is happening. That's all research still in progress. I know that some of you are going to go into that direction, so wow, let me know. There is, however, something known about cortisol and the whole link to stress. What I said earlier with dogs possibly being taken away from their mum and their siblings very early on and way too young, which can cause general stress. Now, the general stress pathway, that would be if cortisol is released by the adrenal glands, just like adrenaline, above your kidneys, the adrenal glands above your kidneys. Now that has to be triggered by something. I think I've had that in an earlier part of your course. Now, I couldn't find the right diagram online, so I'm sorry, I had to just take a picture of your textbook here for this one, because it really seems to go through the pathway quite nicely as to what is going step by step from the brain regions. Literally, you've got the involvement of the hypothalamus and the pituitary, and then the release of literally that pathway, the cascade you're going to get from first a nervous receptor the receptor, that impulse, and going to the hypothalamus either deciding more or less needs to be released. The pituitary then communicates with the adrenal gland that more or less cortisol should be released, which is then the stress hormone. I'm not going to try to reinvent the wheel here. I have found an amazing video for you to watch on that axis, that cascade, the HPA axis, hypothalamus, pituitary, and then adrenals in that axis. Please, please, please either go on YouTube and click on that link or type in 2-minute neuroscience HPA axis. You'll find it. It's just a straightforward video and it talks you through it in 2 minutes across as to how the stress then signaled to the hypothalamus, then signaled to the pituitary. You will need to know the in-betweens. You will need to know, let me just go back to that diagram. You need to know the corticotropin releasing hormone and the vasopressin for the sort of decreased response. So increased and decreased information going to the adrenal glands, make more, make less cortisol. And obviously the question with epigenetics is, at what point do we have which hormone production possibly inhibited by epigenetic changes? So now, if background levels of stress can cause somebody to therefore be constantly overstressed, that normal feedback loop that you've got to, if there's more cortisol, it would actually close back down. It would shut down the release of cortisol. And you would, because that feedback loop to go back into negative feedback and release less cortisol, if that's overridden somehow and you get more and more and more and more cortisol, you will get constantly stressed humans. That might be a reason why individuals who have been subjected to huge stress levels as children, who've had been subjected to abuse in their childhood years, might have higher levels of cortisol all around and all along, and therefore more likely to develop that list of conditions that were on an earlier slide. So just going back a little bit, in case you've got an override, this negative feedback from the cortisol to the receptors that would then tell the hippocampus, right, we've got cortisol, we don't need to produce any more. That feedback, if that is no longer being recognized as a negative feedback loop, and you'll just keep continuing to produce and produce and produce cortisol because of some epigenetic change, possibly children that have been abused will, because of the higher levels of cortisol, struggle with more stress and therefore get that. That might be a possible mechanism, not yet known. Go into it, research it, and see if you can maybe stop some of that. Right.